So Corey completed his PhD in pharmacology in Stephanie Borgland's lab at the University of British Columbia at the Hodgkins Brain Institute in Calgary, Alberta. And there he examined the modulation of VTA dopamine neurons by the neuropeptide orexin. Corey is a postdoctoral fellow now in Adam Carter's lab at the Center for Neuronal Science at NYU, where he's studying anatomical tools, using anatomical tools and slice electrophysiology, optogenetics, and behaviors. He's studying the nucleus accumbens circuitry. And the title of his talk is Afferent Control of Local and Long-Range Circuits in the Nucleus Accumbens. Thank you, Corey.
Thank you, Veronica, for the introduction, and Rebecca and the rest of the organizers for putting this symposium together and for inviting me to talk today. Let me just share the screen, which I think we've worked out how to do it. And since I can't see you guys, can you confirm that you can see my screen and hear me okay?
Yes.
Excellent. Yes, perfect. Thank you. So today I'm going to talk to you about some work I've been doing examining how long-range inputs contact and regulate the activity of various cell types in the nucleus accumbens. Now, this is a part of the brain that I'm sure is familiar to most people here, and it's been long examined for its role in the learning and the expression of flexible motivated behaviors, by way of the fact that it is strongly implicated in neuropsychiatric disorders. The aspect of the accumbens that I'm going to focus on today is in its role as the gateway to the ventral stream of the basal ganglia, which consists of a series of subcortical nuclei that participate in the selection and execution of adaptive behaviors. I refer to it as the gateway because the accumbens is the primary input region to the system, where information from cortical, thalamic, amygdala, and hippocampal inputs can be integrated and used in the transition towards the execution of goal-directed actions. Now, most of the work I'm going to talk about today is focused on this medial shell region of the nucleus accumbens, but it's worth noting that the accumbens itself consists of at least 3 different subregions: the core, the medial, and the lateral shell, and that in all of them, both excitatory inputs and the output neurons of the accumbens themselves are subject to modulation from dopamine ascending from the VTA. Now, within the accumbens, these inputs get integrated onto GABAergic medium spiny neurons, or MSNs, as I will refer to them throughout the talk. Now, MSNs are the principal cell type in the accumbens and represent the output neurons of this region. And throughout the accumbens, they are segregated into 2 broad populations, those that express the dopamine type 1 receptor or the dopamine type 2 receptor. And so you can visualize this here in a confocal image of a coronal section of the nucleus accumbens, which I took from a D1 TD tomato D2 EGFP double transgenic mouse, where I've labeled all neurons with a new antibody. So essentially what you're seeing are D1 MSNs labeled in red and D2 MSNs labeled in green. And one of the first things I did when I joined Adam's lab was to perform an analysis of this mouse in order to confirm the size and the distribution of these 2 MSN populations within the nucleus accumbens. What I'm sure you can appreciate from this analysis shown here is that D1 and D2 MSNs make up the vast majority of all neuronal cell types. In this case, they've roughly worked out to about 90%, but with most of them expressing either D1 or D2 receptors, but very few expressing both receptor subtypes. Now, the precise mechanisms by which D1 and D2 MSNs promote behavior is quite complex, and it's constantly being updated. But what we can take away from the literature is that it seems clear that differential activation of these cells has strong implications for nucleus accumbens function and nucleus accumbens dependent behaviors. As just one example of this, D1 MSNs are preferentially activated by addictive drugs and may encode information about drug associations that can drive relapse. With that being said, MSNs are not the only cell type in the accumbens. They are surrounded by both cholinergic and GABAergic interneurons, with the latter coming in many varieties. So what I'm showing you here are, again, confocal images of the accumbens now, but also expressing markers for cholinergic interneurons, parvalbumin positive interneurons, or somatostatin interneurons, all intermixed amongst D1 MSNs. Now, these neurons will come into play more in the second part of my talk, but now all I really want you to take away from this is that although they are relatively few in number in comparison to the medium spiny neurons, they serve as important local modulators of NAC output and function. But an important point here that I need to make that is critical to the ideas I will present to you today is that there is no significant source of local excitation in the nucleus accumbens. And so because of this, the activity of medium spiny neurons and the output of the region itself is thought to be tightly deregulated by long-range excitatory inputs. And although we have known this for some time, we are very much in the early phases of understanding the detailed cellular and synaptic architecture of the system and how its circuits are recruited and modulated in order to promote adaptive and pathological behaviors. So therefore, the foundational idea behind the work I'm going to present to you today is that in order to understand the role of the accumbens in behavior and then in disease, it's critical that we define the synaptic structure of the system. So with this in mind, I'm going to talk to you about two things today. The first, I will tell you about some work which I published about a year ago now, which examines circuit-specific connections and cocaine-evoked plasticity in the nucleus accumbens medial shell. And then I will transition to some new unpublished work which examines how long-range inputs engage the local circuit in order to modulate cholinergic interneurons. So given that differential activation of D1 and D2 MSNs is important for nucleus accumbens dependent behaviors, and that activity in these regions are tightly regulated by long-range excitatory inputs, our lab set out to determine how different inputs target these two populations of MSNs. We do this by pairing whole cell patch clamp electrophysiology with optogenetics. So this involves AAV injections of channel rhodopsin carrying viruses into the input structures of the accumbens. And so for the first part of this talk, I'll actually focus primarily on the ventral hippocampus and the basolateral amygdala, which are two regions of the brain that our lab has previously identified as being important for drug-evoked behavior and plasticity. So you can see here with example injections into the hippocampus and the amygdala that we get dense labeling in both cases in the nucleus accumbens and in particular in the nucleus accumbens medial shell. We then examine synaptic responses by making sequential paired recordings from neighboring pairs of D1 and D2, or in the work I'm going to present today, D1 positive and D1 negative medium spiny neurons, using D1 negative as a proxy for D2. And we evoke glutamate release with wide field illumination and measure light evoked currents in voltage clamp. And this method of recording pairs of neurons is effective because the absolute amplitude of these currents is ultimately dependent on the number of activated fibers. And so by comparing neighboring pairs of neurons at the same depth in the tissue and within the same local field within the slice, it allows us to control for things like the variability that could arise by location of the cell within the tissue, or from different levels of channel rhodopsin expressed either between slices or between animals. And what we've determined is that while D1 and D2 MSNs receive overlapping inputs, there exist intrinsic biases in the connections and the strengths of these connections. So for example, if we look here where we're isolating ventral hippocampal inputs onto neighboring pairs of D1 positive and D1 negative MSNs, I'm sure you can appreciate that the amplitude of both AMPA receptor recurrence recorded at -70 millivolts as well as NMDA receptor currents recorded at +40 millivolts, in both cases are stronger on the D1 relative to the D1 negative MSN. And so this suggested to us that synaptic connectivity in the medial shell is cell type specific and that we can observe differences between D1 and D2 neurons. That being said, when we perform similar experiments, in this case for amygdala inputs onto these two populations, we find no intrinsic bias in the strength of their connectivity, with the amplitudes of these currents being equal again for both AMPA and NMDA receptors. And so together, this suggests that synaptic connectivity is both cell type specific and input specific, with important differences emerging between D1 and D2 MSNs, but also between the hippocampus and the BLA. We also know that these synapses can undergo experience-dependent plasticity. For example, there's a wealth of literature linking synaptic adaptations in the nucleus accumbens to behavioral phenotypes in animal models of drug addiction, and that this arises at least in part due to drug-induced increases in dopamine levels in the nucleus accumbens medial shell. And so we examined if these connections can undergo experience-dependent plasticity by using a cocaine sensitization paradigm. To test this, we wean pairs of sex-matched litter mates, which undergo all surgical, behavioral, and recording procedures on the same days. And within each pair of mice, one mouse is given repeated access or repeated exposure to cocaine in an open field, while the other receives daily injections of saline. And as you would expect, cocaine-treated mice display this increase in locomotion with each subsequent dose of the drug. Mice are then designated to either go through a cocaine challenge experiment or set aside for slice physiology recordings. And if we first focus on the behavior mice, you can see here that as expected, mice who have previously experienced cocaine injections showed this stereotype sensitized response in terms of increased distant travel to a cocaine challenge injection relative to saline controls. And if we then look at what happens in the slice to the synaptic connections here, what we've seen is that, like synaptic connectivity, cocaine-evoked plasticity is both cell type and input specific. So you can see here that in cocaine mice, we see a dampening of the strength of hippocampal inputs on D1 positive MSNs, so that the amplitudes are equivalent in size and very similar to what we see at neighboring D1 negative MSNs. But interestingly, at the same time point, if we now isolate amygdala inputs, we actually see a very different response, where we see a selective potentiation of currents onto D1 MSNs, again now relative to their neighboring D1 negative neurons. And so together this gave us an idea of how these circuits are wired up and how these connections can be specifically altered by cocaine exposure. But when I joined the lab, it became clear that recent findings had further complicated our understanding of these subjects. So the activation of D1 and D2 MSNs have been shown to drive different patterns of brain-wide activity, and this is thought to occur via the fact that they have non-overlapping projections outside of the striatum. So the dorsal striatum, we would interpret this as connections from D1 MSNs making direct projections to the output nuclei of the basal ganglia and the substantia nigra and the internal portion of the globus pallidus, and D2 neurons doing so indirectly via projections to the external globus pallidus and the subthalamic nucleus. And an equivalent circuit was thought to exist in which D1 MSNs project directly to the VTA, while D2 MSNs do so indirectly via the ventral pallidum. And so in these images I'm showing you here, where I've injected a fluorophore carrying virus into the medial shell of the nucleus accumbens, we can see axon labeling in both the ventral pallidum and the VTA, and we would interpret this as this axon and the pallidum being by way of viral expression in D2 MSNs, and this axon labeling in the VTA by way of viral expression in D1 MSNs. But what's interesting is when you do the same experiment now in a D1 cream mice injecting a Cree-dependent virus, we see the same pattern of axon labeling, with axon present in both the ventral pallidum and in the VTA. And so this demonstrates that this dichotomy of direct and indirect projections does not hold in the nucleus accumbens, where D1 and D2 MSNs can overlap in their projections, in particular to the ventral pallidum. So given this newfound understanding of nucleus accumbens circuits, it seemed important that we refine our understanding of the synaptic connections in the medial shell. And as a first step to this, I aim to better characterize the cell types of the accumbens by looking at whether or not these projections were the product of collateralizing axon or the existence of multiple subpopulations of D1 MSNs in this region. So I tested this using a dual retrograde tracer approach, in which I injected two different colored colorotoxes, shown here in green for injections into the ventral pallidum, or blue for injections into the VTA, and we've done this in D1 TD tomato mice, so in all cases we can identify both D1 and D1 negative medium spiny neurons, and then cut coronal sections and took high resolution confocal images of the nucleus accumbens as shown here, which allow us to simultaneously quantify two things. One, the overlap between cholera toxin and TD tomato, which gives us an idea of what proportion of the projections to the ventral pallidum and the VTA arise from D1 MSNs, and two, the overlap between the two cholera toxins, which allows us to compare to what extent these are the product of individual neurons or neurons with collateralizing axons. And what we found is, as you would expect, the vast majority of all neurons projecting to the VTA consists of D1 MSNs, while the neurons projecting to the ventral pallidum was roughly a 50-50 split between D1 and D2 medium spiny neurons. Interestingly, when we compare the total cell count across these mice, and this was done in 5 separate mice, we see roughly equal numbers of neurons projecting to the VTA or the ventral pallidum, but importantly, very few neurons actually express both cholera toxins and therefore projected to both regions. In total, this was about 7% of all neurons across all these mice. And so this indicates that D1 projections to the VP and the VTA arise from largely distinct cell populations. I also compared the morphology and intrinsic properties of these two populations, but found no major differences between them. Although I do think in the future it will be interesting to adopt methods like RNA sequencing to further compare and contrast these two populations of MSNs. So if we now revisit our sort of simplified circuit model, the nucleus accumbens, I can confirm that these projections to the ventral pallidum and to the BTA don't consist of D1 MSNs that collateralize in both regions, but rather of different subpopulations of MSNs that target either the VP or the BTA. But as I showed you earlier, we know that the long-range inputs to these cells have both input-specific and cell-type-specific connectivity patterns. And so we next assess whether or not these subpopulations of neurons are differentially contacted by hippocampal and BLA inputs, with the goal of determining if connectivity was dependent on the cell type, so D1 versus D2, or the projection target of D1 MSNs. And so we repeated similar experiments to those I described earlier, but now recording from projection identified D1 MSNs by way of cholera toxin injections into either the BTA or the ventral pallidum, and all of this again was done in D1 TD tomato mice. So if we first look at the hippocampal inputs onto neighboring pairs of VTA projecting D1 MSNs and D1 negative MSNs, what you can see is that for both AMPA receptor and NMDA receptor currents, that they are larger or biased at these D1 relative to neighboring D1 negative MSNs. And I've quantified that a few ways here. In the middle is the absolute amplitude of these currents, where each gray line represents an individual pair within the total sample. And now on the right, as an amplitude ratio, where essentially any number above 1 is reflective of a bias towards this D1 VTA projecting neuron. Now this bias is consistent with what we see when we record from pairs of non-retrogradely labeled D1 positive and D1 negative MSNs, as I showed you earlier. But interestingly, it's not what we see when we make similar recordings, but now isolating ventral pallidum projecting D1 MSNs. You can see here in the average traces, as well as in both summary graphs, that the average amplitude is very similar between VP projecting D1 MSNs and neighboring D1 negative MSNs. And so this indicates that only a subset of D1 MSNs are preferentially targeted by the hippocampus, and it's those that project to the VTA. We performed similar experiments for BLA inputs, and as in the non-projection defined samples, you can see here that we observed no obvious bias for any combination of cell types. And so with this information, we can build on our working model of the nucleus accumbens circuit, now with the understanding that hippocampal inputs seem to be selectively biased at synapses onto VTA projecting D1 MSNs, and that the BLA shows no preference for any cell type. And it highlights that projection target is an important determinant of how these cells are wired up in the nucleus accumbens. But as I showed you earlier, we know that cocaine exposure fundamentally rewires circuits in the medial shell, and that these effects occur predominantly at D1 positive MSNs. And so my next aim was to determine if drug-evoked plasticity occurred at all D1 positive MSNs or only those with a specific projection target, which really comes down to the question, does plasticity depend on cell type or on projection target? As before, I did this in mice that had undergone cocaine exposure in a sensitization paradigm. So if we first focus again on hippocampal projections to D1 VTA and D1 negative MSNs, you can see here that in saline control mice, we can reproduce the strong bias of the hippocampal input onto the VTA projecting MSN. And it was super exciting to see that after cocaine exposure, this bias is completely lost. The average current amplitudes are now overlapping for both AMPA receptors and NMDA receptors. And you can see this shift in the ratio of down to about 1. So this suggests that we're seeing robust plasticity at hippocampal to VTA D1 MSN synapses. When we then turn our attention to pairs of VP projecting D1 MSNs and neighboring D1 negative MSNs, you can see here that in saline-treated mice, we again reproduce this result of seeing no preference. And interestingly, these synaptic connections are unchanged or unaltered after cocaine exposure. And so this indicates that both synaptic connectivity and cocaine-evoked plasticity of hippocampal inputs occurs selectively at VTA projecting D1 MSNs. Lastly, we performed equivalent experiments for BLA inputs, which I'll remind you were selectively potentiated at D1 positive MSNs after cocaine exposure. And given that hippocampal plasticity occurred at VTA projecting MSNs, when I started these experiments, I was fairly convinced I would also see plasticity of the BLA input onto the same populations of neurons. and that this would represent a really important shift in the dominant input onto this projection to the VTA. But when we examine the synaptic connections of BLA inputs to these pairs of neurons, what we saw was that we actually saw no change at the strength of what we actually saw was no change in the strength of the BLA connection onto the D1 projecting VTA MSN. You can see here in both saline and cocaine mice that there's no change in the relative strength of these inputs after cocaine exposure. However, when I made similar recordings from pairs of VP projecting, when I made similar recordings this time isolating ventral pallidum projecting MSNs, we can see that there's a really big increase in the strength of these connections from the BLA, in that they are now heavily biased onto the D1 VP projecting MSN. And again, you can see this for both AMPA receptors and NMDA receptor currents. And so this indicated that plasticity of BLA inputs was specific to ventral pallidal projecting D1 MSNs, and that after cocaine exposure, different forms of plasticity occurred for both hippocampal and BLA inputs, and that these effects were highly dependent on the projection target of the medium spiny neuron. And so if we return to our simplified model of the nucleus accumbens circuit, what I've shown you here is that there are multiple subpopulations of D1 MSNs that can be identified by their projection target. So here I focus on the ventral pallidum and the VTA, but recent work from the McNally lab and Christian Lucher's group have shown that a third population exists that projects to the lateral hypothalamus and seems to have unique behavior roles. I've shown you that synaptic connectivity of long-range inputs to these neurons is both input and projection target specific, and that after cocaine exposure, there was a complex reorganization of inputs at these neurons. And I think it's worth mentioning that this reorganization would have been missed had we pooled hippocampal and BLA inputs or D1 and D2 MSNs. And so I think it highlights the need to consider the specific circuits involved when examining how cells and synapses in the accumbens, but likely in any region of the brain, are altered by experience. Beyond that, these results allow us to make predictions about how the hippocampus and the BLA can activate different populations of MSNs and how these dynamics change after exposure to cocaine. But it doesn't tell us about when these cells or inputs themselves are active. And moving forward, we hope to expand on this work by examining these types of questions with the use of fibrophotometry and in vivo physiology with neural pixel probes, which myself and others have been setting up in the lab over the past year. So having examined some of the connections between excitatory inputs and medium spiny neurons, I then transitioned to look at how they can affect other neurons in the local circuit, which in turn may regulate the activity of MSNs. And I focused specifically on cholinergic interneurons, which amongst other things have been shown to regulate aversive, attentional, and motivational processes, and to play important roles in synaptic plasticity, conditioned learning, and action selection. So what you're looking at here is a coronal section of the striatum from a chatback EGFP mouse with cholinergic interneurons labeled in green. I'm sure you'll notice two things right away. One, although they are present throughout the striatum, they're very sparse and very few in number. That being said, they have profound influence on striatal function by way of their ability to modulate multiple nodes in the striatal circuit. And they do this by both direct and indirect mechanisms via a wide range of pre- and postsynaptic cholinergic receptors. So for example, cholinergic interneurons can have direct effects on MSNs through the activation of muscarinic acetylcholine receptors. They can drive feed-forward inhibition onto MSNs by way of nicotinic acetylcholine receptor-dependent spiking of GABAergic interneurons. And there have been well-documented effects for acetylcholine-dependent modulation of both presynaptic glutamate and presynaptic dopamine release. Now, when it comes to the activity of cholinergic interneurons, it is important to note that although they are tonically active, they show phasic activity patterns that are often marked by characteristic pauses in firing. These can take the form of simple pauses, but also pause burst and burst pause sequences. And these patterns of activity have major behavioral implications in that they often occur in response to salient or reward predicting cues. And the Lucher lab has nicely demonstrated that optogenetic induction of pauses, specifically in these cells in the nucleus accumbens, is sufficient to enhance stimulus outcome learning. Now, looking in the other direction, cholinergic interneurons are also transiently activated by cues that discourage motivated behavior. And Kate Wassum's lab recently demonstrated with optogenetics that activation of these cells is sufficient to prevent behavioral action. And so given that the activity patterns of cholinergic interneurons shift with behavior, it's critical to understand what controls their activity. Now, in the dorsal striatum, these neurons are regulated in part by long-range excitatory inputs from the cortex and the thalamus. but the specific excitatory inputs to contact these neurons in the nucleus accumbens is not known. And so in order to understand how afferent inputs regulate their activity in the striatum, we first aim to identify which excitatory inputs actually target these neurons. And we did this by mapping brain-wide monosynaptic inputs to cholinergic interneurons in the nucleus accumbens medial shell using a conditional rabies approach, and this time in chat-crete mice. So this work was largely led by Emily Jang, who's a talented MD PhD student in the lab. What Emily did was first to inject the 2 starter viruses into the nucleus accumbens medial shell. She then waited 5 weeks for expression and finished the experiment by an injection of the G-deleted rabies into this region. And this approach yields starter cells in the medial shell, which are labeled here with white arrows. And the virus will then go from these starter cells, jump back to synapse, and label neurons throughout the brain that project to these specific neurons. So Emily then cut coronal sections of the entire brain and quantified the distribution of input neurons. And what we found was that although there were input cells across many brain areas, the largest density of them were clustered in 3 brain regions, 2 of which I'm showing you here, the midline thalamus and the ventral hippocampus. This was very evident when you look at the overall quantification of input cells throughout the brain, and interestingly, it occurs in the absence of dense labeling in other regions like the prefrontal cortex or the amygdala, which we know to be prominent excitatory inputs to the accumbens. Now, because the midline thalamus consists of multiple subnuclei, we further assessed the distribution of these neurons within midline thalamus and determined that they were primarily located in the peritoneal and the paraventricular nucleus of the thalamus. Now, the third major input we identified was actually the ventral pallidum, which I'm showing you here in this gray bar. Now, unlike the hippocampus and the thalamus, the VP is actually a mixed region made up of both glutamatergic and GABAergic projection neurons. And although we have not tested it directly yet, work from Megan Creed's lab and Tom Nasco's lab has strongly suggests that it's actually the GABA neurons in this region that project back to the ventral pallidum and not the glutamatergic neurons, which seem to target regions more like the VTA and the habenula. And because of this, although this is a really interesting potential input to these cells, for the rest of the talk, I'm actually going to focus primarily on the hippocampus and the thalamic inputs. So in terms of excitatory inputs, this showed us that like the dorsal striatum, cholinergic interneurons in the medial shell receive inputs from a thalamic nucleus, the PBT, and a cortical-like region in the ventral hippocampus. And so having identified these inputs, we next assess what effect they have on the activity of patterns of cholinergic interneurons. And we did this by, again, injecting channelrhodopsin into these input regions, in this case showing for the PBT or the ventral hippocampus, which of course can yield dense axon labeling within the nucleus accumbens medial shell. We then performed whole cell current clamp recordings from GFP-tagged cholinergic interneurons while monitoring spontaneous action potential firing from these cells. And so for the experiments, what I do is actually run each individual neuron through 100 trials, which is depicted here in a few different ways. On the top is an overlay of each trace for all 100 trials from a particular neuron. Below that is the histogram and raster plot of all action potentials aligned across time for that given cell. And at the bottom is a plot of the average spike rate from all neurons in this experiment. And all these plots are aligned to the time of stimulation, which occurs here at the 0-second mark. And for these experiments, it's actually a single pulse, a 2 millisecond single pulse, a blue light, which we use to activate PBT, or as you'll see in a second, hippocampal terminals within the accumbens. And so what I hope you can appreciate from the data I'm showing you here is that brief activation of this thalamic input induces an acute increase in firing aligned to the time of the light stimulation, which quickly returns to baseline once the stimulation turns off. Now, this response is very different from what we see when we do the same experiment, this time activating hippocampal inputs. Instead of this acute increase in firing, what we actually see is a very pronounced pause in firing of the chat interneuron. To highlight how robust this is, if you look here in these, again, this overlay of 100 traces or in the histogram or raster plot, we basically don't see any spiking whatsoever in roughly this 60 to 70 millisecond time period after the light stimulation. Now, in some cells, this pause in firing was preceded by a small increase in spike probability, which is very much akin to the sort of burst pause sequences I described earlier, but this did not happen in all cells, and so we only see a small little peak here prior to this extended pause. And so we've now identified these two primary excitatory inputs to these cholinergic interneurons, and we've shown that they can bidirectionally modulate the firing of these cells. And so we next sought to determine by what synaptic and circuit mechanisms they may underlie this differential modulation. And we were particularly intrigued by the hippocampal evoked pause in firing. Now, as I mentioned earlier, these pauses are very important for nucleus accumbens dependent behaviors. And because of that, a lot of work has gone into determining their cause. And they have been linked, frankly, across the striatum to many different things. Now, this is a long list, so I won't spend too much time going through all the individual examples, but suffice it to say they've been linked to things like the activation of dopamine D2 receptors, by the projections of long-range GABA inputs, for example, from the VTA, and through the activation of intrinsic conductances such as IH or the delayed rectifier potassium channel IKR. But with the data I'm going to show you next, what I hope to convince you of today is that the hippocampal evoked pause we observed is likely through a completely different mechanism. So to get at this, what we first did was starting by looking at the synaptic connections between the thalamus and the hippocampus onto cholinergic interneurons. And so to do this, we performed similar injections of channelrhodopsin into these two regions, and then measured evoked, excitatory, and inhibitory postsynaptic currents in cholinergic cells. And so if we first look here at thalamic inputs from the PBT, what you can see is that this brief activation of the terminals, once again, elicits relatively small EPSCs recorded at minus 60 millivolts, and equally matched in amplitude iPSCs recorded at plus 20 millivolts. Now, these iPSCs are slightly delayed from the onset of the EPSC, which suggests to us that they are actually the product of disynaptic feed-forward inhibition and not direct inhibition from the thalamus itself. Now, although these currents may seem small, we don't think this is because of missed injection sites or weak overall inputs to the medial shub, because when I've made, although I'm not showing you the data here right now, when I've made recordings from neighboring MSNs in these same slices, we do see very large EPSCs and iPSCs, as others have shown in the past. And so we think this targeting is specific to the connections onto cholinergic interneurons. Now, in contrast to the BBT, when we activate hippocampal axons, we can see here's a very different response. We again see these relatively small EPSCs recorded at minus 60 millivolts, but very large and robust iPSCs recorded at positive 20 millivolts, suggest a far more robust recruitment of local feed-forward inhibitory circuits by the hippocampus relative to the PBT. And so to ensure that these iPSCs were in fact disynaptic, we made recordings in the presence of tetrodotoxin and 4-aminopyridine to isolate monosynaptic connections, and saw that the relatively small iPSCs remained intact while the EPSCs remained intact while the iPSCs were completely abolished. And so if we put this together, you can see that excitation and inhibition were relatively balanced for thalamic inputs, but highly biased towards inhibition in the case of the ventral hippocampus, and that this was due to the recruitment of local feed-forward inhibitory circuits. And so we therefore hypothesize that this robust feed-forward inhibition may underlie the hippocampal evoked pauses in firing we observed when we recorded in current clamp. And so to test this hypothesis, we returned to the current clamp experiments, but now recorded with the inclusion of gabazine or MBQX in the bath solution. And what we anticipated was that in the presence of gabazine, we would no longer see hippocampal evoked pauses in firing. And that is exactly what I'm showing you here. You can see that this pause in firing is completely abolished in the presence of gabazine. In fact, that it actually reveals a fairly strong excitatory component that is normally included by the presence of inhibition. And as you would predict, both the pause and this excitation effect are blocked when we record in the presence of MBQX, which would block both direct synaptic connections onto the cholinergic interneuron, but also the recruitment of feed-forward inhibition. And so together, these results indicate that the PBT and the hippocampus differentially engage both mono and disynaptic circuits to modulate cholinergic interneurons. And importantly, that the hippocampus can drive a pause in firing through the recruitment of feed-forward inhibition. And so we next aim to determine the source of feed-forward inhibition, which is an important question because, as I mentioned earlier in the talk, the accumbens is composed almost entirely of GABAergic cells, including medium spiny neurons and multiple subtypes of GABAergic interneurons. And so we performed a series of experiments in order to determine which cell types were being engaged by the hippocampus in order to drive feed-forward inhibition. And these experiments were done along with Samantha Scudder, who was a former postdoc in our lab. So one example of these experiments is shown here. In this case, we're taking a PV2A Cree pup from breeders, a PV2A cream mouse, which is crossed with a chat-EGFP mouse in order to be able to label PV interneurons in these cells. And so what I've done is injected a Cree-dependent fluorophore into the medial shell, and again, the same channel rhodopsin construct into the hippocampus. So you can see here now in these mice, we have in pink or magenta, the PV-labeled neurons, which are interleaved amongst the chat interneurons. And in these mice, we then made sequential paired recordings where we made a voltage clamp recording from a cholinergic interneuron in order to record iPSCs, and a current clamp recording from a PV interneuron in order to look at the firing or action potential firing in the cell. And for each pair of neurons, what we did was actually run through a series of light intensities, making sure that the first was subthreshold, and then increase the light intensity from there in order to get iPSCs of different amplitudes. And you can see this here in these voltage clamp recordings in the cholinergic cell as the amplitude goes from nothing and increases as the traces go from light gray through to black. When we look at the PV interneuron from this specific pair of neurons, you can see that, again, the first stimulation intensity is actually subthreshold. That was subthreshold to yield an iPSC, is also subthreshold to spike the PV cell. But as we ramp up the intensity of the light to recruit more and more to better activate the hippocampal terminals, we start to see robust and very reliable firing of this PV interneuron. Now, we performed similar experiments to this for both pairs of D1 MSN and chat neurons, but also somatostatin and chat interneurons. And this is summarized here. And what I hope you can take away from this is that while both all PV, SOM, and D1 MSNs are capable of firing with stimulations that yield iPSCs in the cholinergic interneuron, early on when the inputs are small, it's really much dominated by these PV cells. And in fact, they're the only cells that fire very reliably throughout all of these experiments. And so this suggested to us that while all three cell types might get engaged by the hippocampus, PV interneurons might be the primary mediator of this feed-forward inhibition. Now, at odds with this hypothesis is that in the dorsal striatum, multiple labs have failed to observe connections between fast-spiking PV interneurons and cholinergic cells. And so we therefore looked for connections between PV cells and chat neurons in the nucleus accumbens, hypothesizing that there may be differences in the local circuit organization of dorsal and ventral striata. And so to test this, what we did was express these, in this case, it's a soma-tagged channel rhodopsin virulent chrome into either the dorsal medial striatum or the nucleus accumbens, and we did this actually in the same mice. And then we performed, again, a series of patch clamp recordings, now recording both in voltage clamp from cholinergic interneuron or a nearby MSN, again, within the same field of view. So we first look at the dorsal medial striatum. What you can see here is that we confirm previous results showing robust inhibition evoked from PV cells onto medium spiny neurons with little to no current seen in the cholinergic interneuron. Now, in contrast, when we do these same experiments in the medial shell, we actually don't see that relationship at all. We see large amplitude iPSCs recorded in both medium spiny neurons and also nearby chat cells. And so if we put this together, what it suggests is that unlike the dorsal striatum, in the nucleus accumbens, PV interneurons can strongly inhibit cholinergic cells. Now, additionally, you'll see here in this summary graph that we also performed similar experiments for both D1 and somatostatin inputs, and we can see in this MSN to chat ratio that in all three cases within the medial shell, we can see robust iPSCs onto both cell types. And so to follow up on this point, we wanted to directly test the contribution of PV interneurons to feed forward inhibition of cholinergic cells. And we did this using an optogenetic suppression experiment. So what we've done here is take a PV2AP mouse crossed with a chat-EGFP mouse, pop from a litter of this cross. So we'll have the ability to, again, simultaneously manipulate PV cells, record from chat cells, and again, we are now injecting channel rhodopsin into the hippocampus, and in this case, it's the inhibitory opsin, RHT, into the medial shell. So we can now simultaneously activate hippocampal inputs while inhibiting PV interneurons. You can see here that RHT is functional in that if we do current step evoked firing in the medial shell, we can inhibit action potential firing when the light is specifically when the light is on, and that if we now drive a spike in this PV cell through activation of hippocampal inputs, we can robustly suppress it in the presence of the yellow light to activate RHT. So what I then did was make voltage snap recordings from these cholinergic interneurons while either activating only the hippocampus or the hippocampal terminals with blue light, or also interleaving trials where we activate the hippocampus and simultaneously suppress cholinergic interneurons with the activation of yellow light. And what you can see here is that ArchT suppression of PV interneurons dramatically can decrease the amplitude of IPSCs recorded in the cholinergic interneuron. And this is in particular in cases where we have low stimulation intensities when the IPSCs are small. So, this is again going through a series of stimulations, and you can see early on that we get about a 40% reduction in the amplitude of the IPSC, which decreases with increased strength of the stimulation, where we think this likely reflects the recruitment of other cell types, which is very much similar to the data I showed you a few slides ago. And so if we put this all together, what I've shown you in the second part of this talk is that the PVT and the hippocampus inputs represent the primary excitatory inputs to cholinergic interneurons in the nucleus accumbens medial shell, that they bidirectionally activate or alter the activity of cholinergic interneurons, and that the hippocampus can induce a pause in cholinergic cells by way of recruitment of local GABAergic neurons, including PV interneurons, which represents a novel mechanism for pausing cholinergic cells that we think is specific to the nucleus accumbens medial shell, again, because we don't see this type of connection in the dorsal striatum. And so to put everything I've put to you, I've spoken to you about today together into kind of one final summary, if nothing else, I hope I've convinced you that there exist important differences between the circuit and synaptic architecture of the dorsal and ventral striatum, and that there is much left to discover about the specific intricacies of the networks within the accumbens. And so given that the theme of today's symposium is the future of dopamine, I think I'll finish by saying that while the work I showed you today focused on the medial shell, I'm currently working on a project that examines subregion-specific connectivity across the nucleus accumbens, and I hope to be able to present that to you in the near future. And so with that, I want to say thank you of course, to the Carter Lab, and in particular, people like Samantha Scudder, Emily Jang, and Castro Manucherry, who have helped out in many different ways in some of the work I presented today, as well as to others at NYU who have helped out with the things I actually do not have the time to talk about today. And of course, to my PhD mentor, Stephanie Borglund, and all of the funding sources. And thank you for listening, and I'll be happy to take any questions.
Excellent, Corey. This was a really thorough talk. You covered a lot of the research in your lab and also from other people in Adam's lab. Very great. There is already one question in the chat, and it's from Rebecca Evans, actually, about whether you could get an inhibitory postsynaptic current as a response of like the firing of one PV neuron. Is this so when you do the pair recordings, can a single action potential on a PV neuron generate an inhibitory response on the cholinergics?
So that's a great question. It's something we haven't actually tested directly. We've done everything with these sequential paired recordings where we do one and then the other. But I would predict you probably could. I mean, PV interneurons are very few in number. And so I think they probably have very widespread projections, both within onto MSNs, but also onto these cholinergic interneurons. They're actually quite hard. That the data I showed at the end with the ArchT suppression, I have to say, is probably one of the hardest experiments I've ever had to do just to get, we had to really try and titrate the expression of the ArchT relative to the channel rhodopsin, and you're trying to hit a small population of neurons that's still, sort of omnipresent throughout the slice, but you're using a viral injection. And I predict if we were to do that direct, let's say patch a PV cell and just inject current into it and record from a chat cell, we probably could. Maybe we'd have to search around for the right combination as with, any paired setup, but I do think we could. I actually predict that because there's so few in number, even though we get this robust inhibition, it's probably not from that many cells. I think the connections are probably very strong. And that kind of comes through in the soma-tagged experiment I showed, where we compared dorsal striatum and ventral striatum, which although we have no control for how many cells, we are not infecting all of them.
Yeah, and with regards to that, if I may interrupt you, because Rebecca's question follows up to that, and I actually had the same question. So very nice that you did the comparison in dorsal striatum. Rebecca, I wanted you to follow up a little bit more on that. But my question is also, their hippocampal inputs are less pronounced also on the dorsal striatum. So I'm not so sure, how can you make that comparison, or what does that mean?
So in that case, we weren't actually, maybe I wasn't clear, we weren't actually stimulating the hippocampus. We were just expressing a soma-tagged opsin into the dorsal medial striatum or into the medial shell of the accumbens, and then activating the PV interneurons directly. And the entire intention of that experiment was just to show that, many labs have shown this lack of connectivity between PV cells and cholinergic cells in the dorsal striatum, but we had this sort of indirect evidence suggesting that this connection did exist, or likely existed, in the ventral striatum. And so that was a way of just comparing that. And it seems that it actually does, is quite a robust connection in the ventral striatum. But we didn't have...
This could do to circuitry then? What do you think are the implications for either the cholinergic interneurons or just the output of the striatum?
I mean, that's a great question, and it's a large question. We could probably talk, the three of us for an hour about that. And that kind of goes back to some work that we've also published, and kind of an overall goal of Adam's lab in general. So Samantha Scudder had a paper from, I think, 2018 now, which looked at inhibition onto D1 and D2 MSNs by way of PV SOM, actually primarily PV and SOM interneurons, and tried to map out how the hippocampus in particular activates these cells. And while the input onto the D1 MSNs is really strong, it's actually a little bit stronger onto the PV cells. Now, what that means from a network perspective and how that comes into play during behavior is a very difficult question to answer. One thing that I have thought about, and I might be biased because of former lab members who have gone to work in the hippocampus, Andrew McCaskill in particular, that we really don't know even all that much about the cells in the hippocampus that are coming here. We're obviously bulk targeting them with the channelrhodopsin injection, but that's not to say that they are the same neurons. We really just don't know. We're just assuming that all of these cells are getting activated by the same axons, but we don't actually know that. And so that's kind of like an important question that applies, I think, to most of the brain. That
yeah, I know. I put you on the spot. But
yeah.
No.
Okay.
I have that question in my head, and I thought maybe you'll help me.
We don't have an answer for it, unfortunately.
Not yet. We have to continue the work then. So actually, two more questions. One is about how old are these animals that you're recording the slices? Are these mature?
They're all adult mice. So we generally wean our mice at about 4 weeks old. I usually wait about a week for surgery, and then it's probably around 3 weeks for expression of the virus. So they're all around 8 to 10 weeks by the time we use them. And that's true for all of the data I showed today.
Excellent. The other question I have brings us back to the hippocampal inputs, and then but your earlier studies with cocaine. Mm-hmm. And, the idea there is that contextual information from the hippocampus, it's coming into the accumbens to be integrated with more reward or other cues associated to reward coming from other inputs to then determine the output of the striatum. However, what you see after cocaine exposure is actually kind of a decrease or a depotentiation of these hippocampal inputs, which in principle was the opposite that I would have predicted. So I wonder if this was also the opposite of what you guys predicted, and how you interpret that decrease in the strength of the inputs rather to more like an ability to drive those D1 cells.
Yeah, that's a great question. And it's something we've, as I'm sure you know, we're not the only ones to look at these types of connections after drug exposure. I don't have a concrete answer, but I will tell you what I think about it. So one important difference is we do these experiments basically 24 hours after the last cocaine exposure. Well, a lot of labs do it like 10, 14, even a month later. And it seems to be a really big difference between acute effects and relapse or withdrawal-induced effects. And I imagine that there is some important regulation going on there. But in terms of interpreting our own data, so we see this depotentiation, let's call it, of the strength of the input seems to occur primarily onto these VTA projecting D1 MSNs. Now, if you try to incorporate other people's work, the Baimel lab has shown that the target of those D1 MSNs is actually, unlike people have previously thought, is actually dopamine neurons, specifically in that medial portion of the VTA, and not so much the GABAergic interneurons, which, you know. So the way I have been interpreting this is that by depotentiating that strength, what you're actually doing is essentially disinhibiting the reciprocal VTA connection that comes back to the medial shell. And that might be important for those kind of acute time point when the reinforcing effects are really dominant. So I kind of conceive of it like that. It's not something we've tested, but I think it's an interesting hypothesis nonetheless.
Excellent. Thank you so much, Corey. That was a really interesting discussion.